A fish with no eyes, responding to light. Not metaphorically, measurably, behaviorally, on camera. 50 meters beneath the limestone plateau of San Luis Potosí, Mexico, a thin stream disappears underground into a system of flooded passages that haven't seen natural light in geological time. The water is cold, the dark absolute. Something lives in it that biology says should be completely blind. Astyanax mexicanus, a small freshwater fish no longer than your palm. The surface version looks ordinary, silver flanks, quick eyes, unremarkable. The cave version has no eyes at all. The orbital sockets are smooth, sealed. The skin grows right over the space where vision should be. Basil Hubbs described the cave populations formally in 1936, collecting specimens from exactly these caves in San Luis Potosí. Scientists assumed what seemed obvious: no eyes, no light sensitivity. Case closed. Damien Moran at the University of Auckland began documenting something that didn't fit the model. Cavefish, fully eyeless, confirmed, were responding to light, not randomly, directionally, consistently. The behavior was logged, repeated, controlled for. It held. The animal had no photoreceptors, no lens, no optic nerve. The hardware for vision, in every classical definition, was absent, and yet the fish was orienting toward illumination. William Jeffrey at the University of Maryland had already identified in 2001 that hedgehog signaling pathways were driving the actual destruction of the eye during embryonic development. The lens cells weren't failing to form. They were dying on purpose. Programmed apoptosis, deliberate, encoded. The cave populations had evolved to dismantle vision multiple times, independently. That last detail matters. Meredith Protas and colleagues published in 2007 that eye loss evolved independently in at least 3 separate cave populations, not one ancestral blind fish, several, convergent, repeated. Something kept responding to light anyway. A specific experiment exists. One tank, one controlled light source, one fish with its remaining sensory systems deliberately blocked. The result no current model in neuroscience can fully explain. The data is published. The explanation is not. That experiment is where this ends. For decades, the answer seemed obvious. Eyes cost energy. In total darkness, eyes offer nothing. Evolution, the logic went, simply stopped maintaining them. This was not a fringe idea. It was the consensus. William R. Jeffrey at the University of Maryland spent years building the molecular case for it. In 2001, his lab published evidence in Science linking eye regression in Astyanax mexicanus to the hedgehog signaling pathway, a master switch in vertebrate development. The lens, specifically, was the trigger, as Yamamoto et al. Confirmed in 2004. The lens cells in cavefish embryos do not simply fail to form. They undergo programmed cell death, apoptosis. The body actively dismantles them. That detail matters. This is not neglect, it is deliberate. The prevailing model was called regressive evolution through neutral mutation and genetic drift. Stripped down, in darkness, mutations that degrade eye tissue carry no penalty. They accumulate. Over thousands of generations, the eye dissolves. Richard Borowski at NYU reinforced this in 2008, mapping multiple genetic loci controlling eye loss across interbreeding surface and cave populations. The trait was real. The mechanism was measurable. Case closed. Except it was not. Here is what the drift model predicted. If eyes were switched off by random decay, the opsin genes should go silent. The protein coding sequences that build photoreceptors should degrade into vestigial noise in a genome that had moved on. Sylvie Retto at CNRS in France was examining developmental gene expression in the early 2010s when the sequencing data returned something wrong. PAX6 and other eye-associated genes were not silent. They were active, not in eye tissue. There was no eye tissue. In other tissue entirely. The 2013 complete genome sequence of Astyanax mexicanus made it undeniable. Opsin genes were not degraded. In cave populations, some were expanding in expression. The genome had not gone dark. It had redirected. So drift explains the empty socket. It does not explain what happened next. If the opsin genes stayed active, stayed functional, after 10,000 years without eyes, something was still using them. The question is where? Here is what the data actually showed, and it broke the framework completely. Masaki Yoshizawa's team at the University of Hawaii didn't set out to overturn a century of assumption. In 2014, working with tissue samples from cave populations of Astyanax mexicanus, they ran standard gene expression analysis looking for what was absent. What they found instead was something that had no business being there. Opsin genes, active, not in the degenerated eye tissue but in the skin. Opsins are the protein-coding sequences that build photoreceptors, the molecular machinery of vision. They belong in eyes. In every vertebrate model, they belong in eyes. Yoshizawa's results, published in Current Biology that year, showed short-wave sensitive opsin families expressing measurably across skin and lateral line tissue in cave populations, not residual, not background noise, measurable expression. The fish had not lost photosensitivity, it had moved it. This was heritable, population-specific, reproducible across multiple cave morphs. Follow-on studies through 2024 confirmed the pattern held, not as an artifact, not as developmental noise, but as a stable genomic signature. The cave populations of Astyanax carry functional opsin machinery distributed across their body surface, encoded in the genome, passed to offspring. Sylvie Reto at CNRS had shown in 2018 the developmental eye genes remain active in non-ocular tissue. Now the picture sharpened. This wasn't leftover machinery from a broken eye. It was a system reorganized and operational. But then comes the question that stops every researcher in that lab cold. If the skin is detecting light, and the genomic evidence says it is, what happens to the signal? The optic nerve runs from eye to brain. That pathway is mapped, labeled, understood. The skin has no equivalent route, no retinal ganglion pathway, no named fiber bundle connecting dermal opsin to behavior. The signal exists. The behavioral response exists. The wiring between them does not appear anywhere in the current anatomical record. So what is carrying the message? Masaki Yoshizawa needed to eliminate every variable but one. His team at the University of Hawaii had already confirmed the lateral line, that network of pressure-sensing hair cells running along the fish's flank, as a serious candidate for light detection. Serious enough to be wrong. So in 2020 they blocked it. Cobalt chloride applied precisely silenced the neuromast cells. The lateral line went dark. Then they put the fish in the tank. One end lit, one end dim. No eyes, no lateral line, nothing left but skin. The fish drifted toward the shadow. Slowly, without urgency, the way something moves when it already knows where it's going. Yoshizawa's team documented the result in Current Biology 2020, blind Astyanax mexicanus, lateral line chemically disabled, consistently oriented toward the dim end of the gradient. The behavior didn't degrade without the lateral line. It persisted, which meant the lateral line wasn't carrying the signal, something else was. That something else had already been confirmed in the genomic data. Opsin genes, the protein coding sequences that build photoreceptors inside vertebrate eyes, were actively expressed across skin and fin tissue in cave populations, not residual, not vestigial noise, expressed, heritable, encoded in the genome, and passed to offspring born in total darkness, generation after generation. This fish did not lose vision. It redistributed it across the entire body surface. Every centimeter of skin a potential receiver. The eye, it turns out, was never the point. It was one solution to a problem the body had already solved another way. But no neuroscience model can currently map the signal pathway. How skin opsin converts light into an orienting behavior. The protein exists. The behavior exists. The mechanism connecting them does not exist yet in any published framework. One tank, one light source, one eyeless fish with a blocked lateral line, and a result that biology has no complete sentence for. The index concludes this. The genome of Astyanax mexicanus contains a functional definition of vision that has nothing to do with eyes, and science has not yet written the explanation to match it.